And our last presenter for the session is Dr. Skip Bohm. Dr. Bohm has over 30 years of experience in laboratory animal medicine with a focus on non-human primates. Over the last 19 years, he has been the attending veterinarian and director of animal care and use programs at 2 National Primate Research Centers, or NPRCs. He currently serves as the Associate Director and Chief Veterinary Medical Officer at the Tulane NPRC and is a member of the NIH NPRC Coronavirus Working Group and co-chair of the Coronavirus Vaccine and Treatment Evaluation Network, COV10, of the NIH Non-Human Primate Research Consortium. COV10 is tasked with developing harmonized research protocols across the NPRC program for NIH's Accelerating COVID-19 Therapeutic Interventions and Vaccines, or ACTIVE program. Dr. Bohm also organized and provided oversight of animal model development for the SARS-CoV-2, rhesus macaque, and African green monkey animal models at the Tulane NPRC. His presentation today is titled Harmonizing Coronavirus Research Across the NPRC's Coronavirus Vaccine and Therapeutic Evaluation Network, COV10. Welcome, Dr. Bohm, and the presentation is now yours.
Thank you for that introduction. I probably don't even need to do this talk anymore as we summarize what COV10 is. But over the last year, the National Primate Research Centers have taken several different approaches to conserving the scarce non-human primate resources at the centers in the face of the need to create therapies and vaccines for COVID. And today I'm going to discuss specifically one of those approaches, which was to create the Coronavirus Vaccine and Therapeutic Evaluation Network, or COV10. Oh, sorry about that. And the first discussions about creating a plan for harmonizing COVID research across the National Primate Research Centers was between the active group at NIH and the National Primate Research Center directors. And for those of you all that are not familiar with the active group at NIH, that acronym stands for Accelerating COVID-19 Therapeutic Intervention and Vaccines. And that group brings together several agencies within the DHHS, including BARDA, CDC, FDA, the DoD, the VA, and the European Medicines Agency. And just as important, representatives from academia, philanthropic organizations, and numerous biopharmaceutical companies were also represented in ACTIVE, which makes ACTIVE truly a public-private partnership. That is tasked to develop a coordinated research strategy for prioritizing and speeding development of the most promising treatments and vaccines. And the word prioritizing is going to come up a number of times during the presentation, and that's because when you have scarce resources, that's one approach to mitigating, is to prioritize the most promising studies using those resources. So the COV10 is a consortium of the 7 National Primate Research Centers, and the overall mission of COV10 is to harmonize research resources and standardize SOPs for COVID research using non-human primates. And while these practices have been going on a long time across the primate centers, harmonization, standardization, they rose to a much higher level with the advent of this pandemic. So the overall goals of COV10 were to conserve and efficiently manage scarce non-human primate resources, which would result in a reduction of the number of animals used, to harmonize COVID research procedures across the NPRC program, which also would reduce the number of animals used by sharing, more effective sharing of data and sharing of controls, and also refinement to laboratory practices in the procedures that we perform in animals. And finally, to allocate COVID research resources to NIH prioritized research projects, and also contributes to the reduction of the number of animals that need to be used in these studies. Many may be familiar, and with the title of this talk, you're probably familiar with the idea that in most years, the National Primate Research Centers barely meet or unable to meet all the requests for non-human primates for research. And this has been the case for some time, even before the pandemic. And in 2018, NIH released a non-human primate evaluation and analysis report, which was developed in collaboration with NIH-funded scientists, some industry private sector partners, and the National Primate Research Center Breeding Colony Management Consortium Working Group. And there were a number of recommendations that came out of those reports, most notably that there was a need to expand existing colonies of rhesus macaques, including both SPF and the expanded SPF colonies by up to 25%. And in addition, there was a recommendation to increase funding to grants supporting those colonies, the P51 Primate Research Center grants, P40 Center grants, the U42 SPF grants, and research resource grants, the R24s, to enable improvements to infrastructure and provide support for expanding colonies to meet demand. And some of these things have been realized by adding additional supplements to these grants, as well as adding construction grants from NIH. So if we've got kind of we're barely meeting the need in a regular year, when a public health crisis comes up, such as COVID-19 pandemic, it only exacerbates the existing mismatch. Sherry Hild and her colleagues at ORIP published a white paper recently in Lab Animal, which just reiterated this, stating that SARS-CoV pandemic has significantly increased the demand for non-human primates, and also for biocontainment laboratory spaces and for skilled personnel to work in these areas. So all of this adds up to knowledge that this was coming, that this was going to be a strain on the system, and it has promoted consideration of strategies for expanding the population of non-human primates used for research and other research resources, and also pointed discussions towards what we're talking about here, which is harmonization across the centers to reduce the number of animals that had to be used. To further the point one more step, NIH published a notice of limited availability for research non-human primates. Again, what it stated was availability of primates will be limited, and that specific programmatic priority settings will be implemented to minimize impact of the shortage, and also talked about prioritizing the most promising studies. And again, this was a little bit unprecedented in that NIH published this notice to put investigators on notice that there were going to be processes put in place for prioritizing animals. And this is the schematic that was created early in the pandemic in anticipation of increased use of non-human primates. And so it's the study prioritization process overview. And to the left of the screen, you'll see the sources of research requests, no different than usual, except in this case, active would be participating in providing requests in what used to be called Operation Warp Speed. And then our regular requests had come from intramural and extramural researchers from NIH and our non-NIH funded researchers, which tended to be quite important in the development of vaccines. So the process was that any entity that wanted to do COVID research had to submit an application through the COVID-19 non-human primate study portal. And then it was reviewed by the expert panel at NIH in association with ORIP. Once those recommendations were made and studies were chosen for priority to be tested, then those were forwarded to the Non-Human Primate Coordinating Center, which is administered at Tulane. And in cooperation with the COV-10 Operations Committee, decisions were to be made as to which centers could accommodate specific studies, had the resources, had the personnel to do those studies. And then the process kind of took over its regular route, which was within the individual primate centers, the Research Advisory Committee would make decisions as to whether resources could be dedicated to these different studies. And at the same time, down at the bottom here, we were conducting non-COVID research. And so the Resource Advisory Committee had to be sure that we could continue to do that as much as possible under these circumstances. And then finally, after all these approvals were in place, studies could begin. So this is really a change in taking the ability of the centers by themselves to allocate animals to studies and had NIH involved in the process. So COV-10 operations group consists of individuals from each of the 7 centers. Each center had 2 individuals in COV-10. One of those was a scientist with expertise in infectious disease research, and the other was an animal resources administrator. And there were 3 co-chairs of the group chosen: Koen van Rompuy from the California National Primate Research Center, Deb Fuller from the Washington National Primate Research Center, and I was representing the Tulane National Primate Research Center. And in addition, we had Angie Birnbaum, who's a biosafety officer. Sherry Hild was our NIH liaison advisor, and John Nylander was the administrator of the program. So one of the first steps we took in the leadership group was to establish subcommittees, and we decided that we needed to have expertise in certain areas to determine which areas of the programs needed to be harmonized or could be harmonized. And as with the COVE 10 overall, all 7 National Primate Research Centers were represented in each of these expert subcommittees. And the function of the committees was to provide information and recommendations to the COVE 10 leadership group to develop a harmonization plan. And after the harmonization plan was developed, it was our idea to keep the subcommittees active because these SOPs, harmonized SOPs and procedures would be, as we learned more and did more research, the SOPs would be informed and improved by that and refined by that. So we needed to keep them plugged in. So this is a list of the subcommittees. I won't go through all of them, but pretty expansive. Subcommittees in each had a chair and a point of contact, which is one of the leaders in the on the COVE 10 leadership group. And we divided the subcommittees into 2 broad categories. One was laboratory subcommittees, which included virus production, virus load, antibody responses, innate and inflammatory responses, and cellular immune responses. The second group was animal-related and other subcommittees. So for the laboratory subcommittees, the first task that they had was to develop a list of assays that were critical for COVID research. And then out of that list, determine which of those could be harmonized. And so, as you're aware, laboratories generally have different practices to some extent, and some of the assays that were really critical, such as flow cytometry, were more difficult to harmonize across centers than some of the other assays. Once they developed that list of assays that were important and could be harmonized, we wanted them to look at developing a list of centralized cores. So by that, I mean, instead of doing the assay at 7 different centers, each center does its own, that there may be some benefit in creating a centralized or using centralized cores either at one of the centers or at perhaps an NIH contracted core that had high throughput, obviously, and was well validated. And then once all that information was in hand, we asked them to create SOPs that could be harmonized across all of the centers. Likewise, the animal-related subcommittees, which included animal pre-screening, sample collection and procedures, sample processing and biostatistics. We asked them to also develop a list of potential SOPs that could be harmonized, and then they wrote those. We set up a pretty rigorous review process through each of the subcommittees, then through the leadership core. And in addition, the National Primate Research Center Rigor and Reproducibility Working Group also went through all of these SOPs for review. Another thing that we were tasked to do is to develop a field guide, and that was a guide that was made available to the public, and was targeted towards scientists who were not familiar with non-human primate research. Because in the early days of the pandemic, each of the primate centers were getting a number of requests, email, phone calls, Zoom meeting requests to participate in studies, and it was clear that a lot of the individuals who were asking to perform the studies hadn't done these studies in non-human primates. So we felt like it would be a good primer for them to have the information that was essentially in all of the SOPs that was available for the public to use. So what we were able to accomplish, obviously lots of Zoom meetings between the subcommittees, the operations, the leadership group, and for a while we were having several meetings a week. We created, reviewed, and revised harmonized SOPs, turned out to be 41 SOPs in total, 252-page document. And you see here a listing of the different categories of SOPs. The highest number of SOPs was in the sample collection, because those are really easy to harmonize across the centers, and how we collected samples from animals, how we inoculated animals, and also we completed the field guide, and I'll talk about that in a minute. We identified centralized cores. Most of these were NIH contract cores for viral production, neutralizing antibody assays, viral load, and genomics. So one of the first things we did before we did the first SOP or identified any laboratory assays was to create a clinical scoring algorithm, and by that we were taking a somewhat subjective physical exam findings sort of process and converting it to a numbered process for scoring. And we used our knowledge of what had gone on in our model development studies, for which were the most important clinical signs that we saw in those studies. And so by attributing numbers, we could compare more effectively across centers. And so we developed this process for that, and it is in use right now. We also developed a template for post-inoculation protocol for timing and the types of samples that we were collecting in our model development studies. And certainly there's a lot of variability in these types of studies, whether you're looking at a vaccine or a therapy, and different timelines after, but this was meant as a starting point, and reflects what the primate centers had done in their model development studies. So as I said, the field guide was developed. It is a representation of all those harmonized SOPs. Talks a lot about selection of animals, species, et cetera, inoculation routes, and then the immunologic assays. And it's available to everybody. You can click this link and find it, or type field guide for SARS-CoV-2, and it's easy to find. And some of the more recent activity that we've had with CoV-10 is we're testing all these protocols and our processes by performing a study at the Yerkes National Primate Research Center. It's an interferon gamma blockade study to induce inflammation and perhaps create a model that would be better for testing therapies. So it's given us an opportunity to test all these cross-center protocols, as well as data sharing, because we have a data sharing center as part of the coronavirus center at Tulane. And so this has given us an opportunity to test that, and so that's ongoing, and so far so good. The other thing that we're talking about recently with a lot of earnest is the post-acute sequelae of COVID, or long COVID research studies. This is a really important and emerging area of research, and some of our findings in non-human primates show that even in the subclinical cases, that these animals have lesions, particularly in the brain and other organs, that could affect an animal chronically. So we want to follow up on that, and in order to follow that, we added a neuroscience subcommittee to the other list of committees, and they are coming up with neuroscience protocols for sampling and processing of tissues. And one of the other big topics that we're talking about with regard to long COVID is how we contain chronically infected animals that aren't shedding. Do they need to be in ABSL3 for months and months on end, or can we reduce that containment? So there's a lot of discussions regarding that. So just to reiterate, a lot of this harmonization and sharing of data reflects positively on our ability to reduce the number of animals that we use. Also, to sharing data, it increases our ability to refine our practices, including all of the procedures that are done in animals. So overall, we've seen the possibility to reduce and refine our practices. And with that, that's the end of my talk.